Welcome, fellow cardiologists, molecular pharmacologists, and science enthusiasts. It's an absolute pleasure to have you here today for this technical explainer. We are looking at what is genuinely a revolutionary leap in precision medicine, the apoquidina 3 compound. Look, if you've been following the ongoing logistical nightmare of managing hereditary cardiovascular diseases, specifically the elusive lipoprotein A, or LPA, you know exactly how desperate we are for a major breakthrough. Well, the rational design of this G4 stabilizing molecule just might be the elegant solution we've been waiting for. So let's dive right into the science. Okay, let's look at the agenda really quickly. We'll cover 1. The logistical and clinical challenge, 2. Molecular mechanism of action, 3. Pharmacokinetics and safety, 4. Molecular simplicity versus biologics, 5. Specifications and SNILS code, and finally, 6. Open science and global access. Let's get to it. Section 1. El desafío logístico y clínico. Overcoming the barriers of LPA management. As specialists, you know the massive hurdle we face in modern cardiology. Managing hereditary cardiovascular diseases without completely overburdening the patient is, well, it's a really tough balancing act. Right now, the gold standard for advanced LPA control relies heavily on injectable biological therapies, things like antisense oligonucleotides or siRNAs, and yes, they work, but they come with huge drawbacks. Poor long-term patient adherence, incredibly complex cold chain logistics, and a massive dependency on centralized hospital infrastructure. In stark contrast, a daily oral small molecule drug, that gives you high adherence, practically zero logistical friction, and ridiculously easy global distribution. The whole goal of apoquidina 3 is to take the absolute power of precision medicine and pack it into the simplicity of a daily pill, totally bypassing the logistical nightmares of traditional injectables. Let's move to Section 2, Mecanismo de Acción Molecular, Post-Transcriptional Modulation via RNA G4. Now here is where the technical brilliance of post-transcriptional modulation really shines. Step 1. The molecule hyperspecifically targets the 5' untranslated region, the UTR, of the ApoA messenger RNA. Step 2, it binds to this region and drastically increases the thermodynamic stability of the RNA G-quadruplex knot, the G4. Step 3, by hyperstabilizing this knot, the molecule creates a physical steric hindrance. Basically, it builds an impassable roadblock that completely stops ribosome scanning and the translational machinery. And finally, step 4, with the ribosome physically blocked, protein synthesis is completely inhibited. Clinically, this approach is projected to slash circulating LPA levels by a massive 85%. It's literally a microscopic roadblock that translates into macroscopic cardiovascular benefits. Section 3. Perfil farmacocinético and seguridad.
Precision engineered for chronic care.
Now, what's really interesting about this next part is how it addresses safety. When we prescribe a lifelong treatment, we absolutely demand strict safety profiles, right? Well, apoquidina 3 is brilliantly engineered as a hepatic prodrug. Normally, the first-pass effect in the liver is a massive headache in pharmacology, but here, it's leveraged as a crucial advantage. You ingest the compound in a pharmacologically inactive state. It then undergoes metabolic activation exactly where it's needed, through that first-pass hepatic metabolism, right in the liver, which happens to be the exact site of LPA production. This cleverly optimizes the targeted efficacy of the compound while keeping systemic exposure to an absolute minimum. Digging into the physical chemical properties, the chemical core is a fluorinated thieno-3,2-G quinoline with a 7-methoxyl group. But the truly spectacular part here is the affinity. A dissociation constant, or KD, of exactly 45 nanomolar, which has been rigorously validated through in silico simulations. Plus, the compound exhibits highly selective targeting, showing clear discrimination for RNA G4 structures over DNA G4. This selectivity is absolutely vital because we want a strictly translational blockade. By completely ignoring DNA, the molecule preserves genomic integrity and prevents any potentially dangerous transcriptional interference. And we can't talk about cardiovascular safety without highlighting this: absolutely zero hERG toxicity. This is a big deal. A lot of G4 ligands fail in clinical trials because they interact with the hERG ion channel, causing severe cardiotoxicity. The authors of this white paper achieved a major engineering feat by strategically reducing the lateral chain to optimize the molecular pharmacophore. They successfully uncoupled therapeutic efficacy from cardiotoxicity. For chronic, long-term cardiovascular treatments, this is a total game changer that ensures absolute safety. Moving on to Section 4, Simplicidad Molecular versus Biologicos, Redefining Precision Medicine. And this brilliantly illustrates a fundamental paradigm shift in the industry's structural philosophy. The current way of biologics, like siRNAs and antisense oligonucleotides, they are massively complex, incredibly expensive to manufacture, and they severely limit patient accessibility. Apoboadina 3, on the other hand, is a small molecule that offers pure synthetic simplicity. It allows us to take precision medicine out of specialized clinical centers and put it directly into the patient's hands at home. It proves without a doubt that an advanced genetic approach doesn't necessarily require the heavy, expensive complexity of today's biological therapies.
Sección 5. Especificaciones y código SMILs. Open data for peer validation.
This white paper isn't just a theoretical pitch. It's an open invitation to the global scientific community. Take a look at this SMILES code string for Apoboadina 3. This is the master key, right here. By publishing this technical protocol openly, any academic institution or independent synthesis lab can immediately run their own in silico simulations, perform molecular docking, and actually replicate the synthesis of the compound to validate its efficacy. No barriers, just open science.
Section 6. Ciencia abierta y acceso global. Democratizing cardiovascular treatments.
So the crucial point is how the Apoboadina project pairs its rigorous medical approach with a highly practical distribution model. It's released under a Creative Commons CC BY 4.0 license, meaning absolutely zero restrictive patents. Because it's a small molecule, its chemical simplicity allows for global decentralized production by generic laboratories right from day one. This completely flips the economic paradigm of modern medicine on its head. It makes an essential cutting-edge cardiovascular treatment economically viable worldwide, directly combating inequality and empowering global health access.
To summarize the social benefit and the massive ethical imperative behind this project, the author Lázara López Paz writes, Hacia una medicina basada en la conciencia y el acceso universal, toward a medicine based on conscience and universal access. It's a powerful reminder that the ultimate goal of pharmacological innovation shouldn't just be clinical efficacy, it has to be equity too.
So, I'll leave you with this provocative question. Is cardiology ready to trade the monopoly of biologics for the equity of open science? It's a deep reflection that challenges both our clinical habits and existing industry structures. The science behind Apoboadina 3 shows that the mechanism is highly effective and incredibly safe. Now it's up to the medical community to validate it and embrace the universal access it provides. I highly encourage all you specialists to dive into the full white paper, run the SMILS code, and see for yourselves. Thanks for joining me for this explainer, and keep questioning the status quo.